Hi, everyone. Hi, thank you for joining us today for the latest edition of the Center for Wildlife Studies Wildlife Chronicles webinars. Just to start, I'll introduce Center for Wildlife Studies. So the Center for Wildlife Studies is an internationally recognized center of excellence in the arenas of wildlife research, conservation, policy, and education. So we used to have talks in our Bangalore office, by leading experts in conservation and wildlife science. But because of the pandemic, we started hosting webinars. All of our past webinars can be found on our YouTube channel. And we've had some extremely interesting topics from snakes and elephants and primates to policy, law, and environmental education. So please do give our YouTube channel a look if you have the time. Today's webinar will also be uploaded on YouTube if you want to watch it later. If you want to support our work, you can use this QR code to donate. And you can also reach us at outreach@cwsindia.org. So with that, I'll just get right into the webinar. So today's webinar is about how we can use evolutionary biology to improve conservation. Conservation, we're trying to protect the biodiversity on our planet. And by understanding by understanding how organisms evolve, how they adapt, and by looking closer at what's going on at a genetic level, we can use that to improve conservation and make it more effective. So we have two extremely distinguished speakers here with us today. So Dr. Patricia Brekke is a scientist at the Zoological Society of London. She studies anthropogenic impacts and the ecological, genetic, and evolutionary consequences of conservation interventions on wildlife populations. She's worked extensively on wild and captive bred bird populations, and she works closely with government bodies, NGOs, and community groups to apply her research. Dr. Praveen Karanth is a professor at the Indian Institute of Science. He specializes in historical biogeography, molecular phylogenetics, character evolution, and population and conservation genetics. His lab works on a wide range of systems, such as langurs, geckos, butterflies, tarantulas, and centipedes, to answer exciting questions in biology using an evolutionary framework. So to start off our webinar, I just want to ask our panelists, how did you get interested in evolutionary biology? And also, can you explain some fundamental concepts to our audience briefly? Patricia, do you want to go first?
Sure. So I guess my interest in wildlife and evolutionary biology started from a very young age. I was a child, I grew up in Colombia, so I was surrounded by wildlife, by hummingbirds in my garden, and me and my sister used to go out and collect insects and snails and arthropods in biscuit tins, which my mother absolutely hated. And then as I grew a little bit older, then I really enjoyed watching wildlife documentaries and kind of decided what I really loved and was passionate about was animal behavior. And so I went to study zoology at university and did a lot of animal behavior courses. And during that degree, I also studied evolution in different forms. And I became really interested in sexual selection, which is, reproduction and how species select mates. And I was really lucky that I got a job as a research technician in Tim Burkhead's lab when I finished my undergraduate degree, and he works on sexual selection and sperm competition in zebra finches. And so I got a really good grounding on what day-to-day research is in evolutionary biology. But when it came to do my own PhD, I really wanted to do something that was a lot more applied and that had a conservation angle. So I kind of merged the evolutionary biology I had learned in my undergraduate degree and during my technical stint as a technician with conservation management. And that's how I then started working, applying evolution to conservation.
Wow. Praveen, what about you?
So actually, even in my case, my interest in ecology and wildlife started when I was quite young. And basically when I was in 8th standard, I attended a nature camp organized by WWF. And I don't know if they do that anymore, So that was a life-changing moment. And then up until then, I wanted to join the Air Force, and then the thing just came along, and I was like, that's it. I want to do wildlife research. So and then I went on to do see, in India, we don't have a bachelor's program in ecology or evolution. So, I had to do something closest to it. And that was the one I could find was bachelor's in agriculture. That there was not much ecology or evolution there either. Anyway, so I went on to then do a PhD in evolutionary biology. And originally I wanted to actually work on cooperatively breeding birds. But when I got there, there was the person I wanted to work with didn't have the funding. Some other faculty had a lot of funding for work on langurs in Asia. So that's how I got involved in the langur project. And turns out there's lot of really interesting questions that needs to be addressed there. Yeah, so I basically work on the mainstay of my lab is phylogenetics. So we build phylogenies using DNA sequence data. And the idea really is that you sequence a particular gene from a bunch of different species for which you want to try and figure out how they are related to each other. And then you look at the variation in the DNA sequence and use that information to infer the phylogeny. And once we have the phylogeny, we then go on to address other interesting questions in evolutionary biology. So yeah, that's briefly what I do.
Thank you. So my next question is for Patricia. For years, you've studied birds in New Zealand, and particularly a threatened bird called the hihi. So can you tell us more about your work on these birds and why it interests you?
Sure. So I started working on hihi during my PhD. So originally my PhD question was trying to look at honey eaters in Australasia, but it turns out the first species I went to look at, which was the hihi, isn't a honey eater. Molecular phylogenetics helped clear that up. And so we found out it was an endemic species to New Zealand and the sole representative of its family. So it's a really unique species. And also it's a really interesting species from a mating system perspective. It has a really complex and unusual mating system, which really makes it a very interesting species to focus on. And New Zealand has a great history of conservation. And so for this particular species, the reintroduction program has started in the 1980s. So there was an incredible amount of information for the species for the whole reintroduction history and establishment of the species from one island to the next. So they established new populations and all that establishment history was known. And also they track every individual from birth to death. So we have an incredible data set that is perfect for asking evolutionary questions that require these really big longitudinal data sets that are normally only available for domestic species or for captive species, very rarely for wild animals, and especially wild endangered animals. So that's why I decided to focus on the hihi.
That's crazy. That sounds like an amazing data set to have.
It really is, yeah.
And the fact that that bird is the only one of its lineage in New Zealand, right? That's
in the whole world, yeah.
Oh, wow.
Yeah.
What about, so Praveen, you've worked extensively on studying the evolutionary relationships of species in the Indian subcontinent. So what have been some of your most interesting findings?
Oh, geez, there have been quite a few. See, when I returned to India in 2005, I was probably one of the few molecular phylogeneticists in the country. Definitely the very few trained molecular phylogeneticists. So there was like so many questions to be addressed using phylogenetic methods. So when we started our work, one of the first things we realized was there was a lot of cryptic diversity. These are species that morphologically look similar, but when you do the molecular work, you realize, hey, you're dealing with multiple species here. So so a lot of cryptic diversity, taxonomy of many of the groups had to be redone. And then when we started looking at higher level patterns, lots of really interesting things emerged. One of them is that we realized that in case of India, we find endemic radiations in many of these groups. And by that, I mean, you have all the species in a particular, in the Indian subcontinent are intimately related to each other. They share an immediate common ancestor. And that means that there was a single dispersal into India and then they radiated. These kinds of radiations you often find on islands, and we are beginning to find them in India, and that was very, very exciting. So I think India is sort of like an island. I've actually written a paper on that. And then we also found something else that was quite interesting. For the longest time, people in India thought that because India was part of the Gondwanan supercontinent, and then it broke apart from Gondwana and merged with Asia. So a lot of people thought that India carried with it many Gondwanan elements to Asia, right? And India's biota is unique because it also has elements from the southern continents. But in due course in time, we realized that with respect to tetrapods, basically vertebrates with 4 limbs, so fishes, amphibians, no, not the fishes, amphibians, reptiles, birds, mammals, basically none of them have Gondwanan origin. They all came into India. So that was a huge revelation, right? And yeah, I mean, there are a lot of other interesting things. We discovered many hybrid lineages recently. We discovered this particular species that was thought to be widely distributed. Actually, it turns out in the Western Ghats, it's a new species, and it has a hybrid origin. And we suspect it reproduces through parthenogenesis, right? So these are some amazing discoveries. There are quite a few others, but it's been an exciting time.
That's amazing. I've actually never heard of these things before, and that's really interesting to find out that India is like an island. So, I would love to hear more in detail about your work. And but in this webinar, I want to talk a little bit about conservation, right? How we can apply evolutionary biology for conservation. So my next question is, in the last few centuries, so many wildlife populations have become threatened because of habitat loss and hunting, and because of invasive species and so many other factors. So, what are the evolutionary or genetic factors involved in this? And, is there anything that distinguishes populations that are threatened? How has their evolution and genes changed because of this, Patricia, do you want to go first?
Sure. So the hallmarks of a threatened species are that they're in small populations, and usually they're very isolated. And this creates a number of genetic risks for these populations. So mutation accumulation is a common one. Also inbreeding, which is probably one that you have heard of most. And inbreeding can lead to inbreeding depression. So inbreeding basically occurs when relatives mate, so brothers and sisters or cousins or parents and offspring. And when this happens, the offspring that they produce generally have a lower fitness, which means that less able to reproduce and less able to survive. And this is called inbreeding depression. So that's one of the issues that small populations face, small populations of threatened species particularly. But also they lose a lot of genetic diversity, and this occurs because, again, they're in small populations. So if 1 or 2 individuals die, they take away a lot of the genetic diversity that is happening in that population because it's so small. And this normally happens through a process called genetic drift, which is a stochastic process, a random process. So all of those issues mean that small populations are more likely to have genetic problems, and also not able to adapt to new environmental conditions or new diseases or new challenges that they face, because they don't have the diversity in the number of individuals that they have to face these issues. For example, the Tasmanian devil, it has a facial tumor cancer that is spread from individual to individual through physical contact during the breeding season. And because the Tasmanian devil has very low levels of genetic diversity, new individuals that are exposed to the facial tumor don't recognize these cells as not being of themselves. So they don't recognize the molecular composition of these cancerous cells. And so then they get the disease themselves because they're not able to fight it off. And that has decimated about 50% of the Tasmanian devil population. So it's had a massive effect on this species. So genetic risks can be a really big issue for threatened populations.
Wow. Praveen, do you have anything to add? And also, do you have any examples?
Not really. So this is more of a population genetics problem, right? What Patricia was talking about, small populations, low genetic diversity. You know, you can also have lethal alleles expressing and, you know, stuff like that. However, you know, phylogenetics has been quite informative in trying to figure out if lineages have lineages exhibit niche conservatism, right? So if you think about say 2 phylogenies, in one phylogeny, say there are, you know, 10 species, and let's say, you know, 5 of them are distributed in the dry zone of India, 5 of them in the wet zone, but they are all interspersed in the phylogeny. And that would basically then mean that this whole clade does not exhibit much niche conservatism. They are able to switch between dry and wet zones, right? Whereas if you have a clade or a group of species branching together, all distributed in the same habitat or ecological zone, you know, or environmental zone, then the ancestor was also distributed in that kind of a setting, right? So the niches of these species are then highly conserved. They might not be able to adapt to changing environmental conditions, right? So if we build phylogenies of various groups and then try and figure out which are the groups that exhibit niche conservatism, such lineages, I think, are more prone to extinction because of climate change or whatever in future. So that is something I think worth doing. We haven't done much of that kind of work here in India, but people are beginning to look into it in other parts of the world.
Group more adaptable than others, like generalists versus specialists kind of a thing?
Sort of, broadly speaking, yes, yeah. Yes. One can, you know, yeah, expand the definition to that, yes. Yeah.
So you can use phylogenies to figure out which ones are more likely to be threatened because they're more, they're found in very specific habitats.
And especially if you have a situation where there's a single lineage, does not have many species in that lineage, and it is highly niche conserved, that lineage then is very valuable because, you know, if you have a few of those species vanish, then that's the end of that lineage, right? Yeah. Yeah.
Do you have any examples from India? I mean, you said not much work has been done, but are there any examples?
No, not with respect to niche conservatism, but evolutionarily distinct lineages. People are beginning to identify in many groups, especially distinct lineages that that are endemic, have very, very restricted distribution. These are the species that we should concentrate because phylogenetically they're unique and they have very restricted distribution. So they are highly threatened, you know.
So my next question is actually is for you only. So we use the word species so often, and you know, we talk about, you know, this species of bird is different from this species, and sometimes these species visually you can't even distinguish between them. So what does species actually mean? How do we know that one species is different from another? And you know, what's the modern definition of species genetically? And also, what is the relevance of this for wildlife management or for conservation?
Yeah, I think that's very fundamental question in evolutionary biology, right? What is a species? Traditionally, we have described species based on morphological characters, right? And the most commonly accepted definition or species concept is the biological species concept, which basically emphasizes on reproductive isolation. Uh, and genetic data has actually helped us, determine if there is reproductive isolation or not. And what genetic data has also helped us is identify cryptic species. So what happens is when you're describing the species, you're using morphological data, and then you're saying that morphologically they're different, so they probably are not interbreeding, they are two different species. So I'm using the species concept idea here to call them two different species. However, we have cryptic species that I talked about earlier, right? Morphologically, they look the same. How do we know they're two separate species? When you do the genetic work, you realize, they are falling into these two very distinct groups, and each group is distributed in a different ecological zone. And when you look more closely, you actually realize they are two different species. What this has done is, so we have basically underestimated the diversity of many of our widely distributed species. So if you have a widely distributed, see, India is very diverse, right? If you go from North India all the way to South India, it is very diverse in terms of climate, habitat, topography. And if you have a widely distributed species across this whole range, and it is not a large animal, uh, it's a much smaller animal, then most likely it is a cryptic species complex consisting of many species. And one can then use molecular data to find out what is the actual diversity in this group. Now, why is this important? Let me go back to the langur work. When I started my PhD, the consensus was there's only one species of, there are multiple species of langurs in India, but there's this one species called Hanuman langur, was divided up into a few subspecies, but it was considered one single species from Kashmir all the way into Sri Lanka, right? Now we know it's 4 different species. After doing a lot of genetic work, morphological work, ecological niche modeling and all that. So we went from a widely distributed common species to 4 different species. And some of these species actually have very restricted distributions, right? So that is where the conservation implication comes in, because earlier you were like, hey, it's the same species that's found over there, but no, it's something different. And it has a very restricted distribution. So some of these, among these 4 species, there's one particular species that has a very not very restricted distribution, but it's not as widely distributed as the rest. So we can then go on to say that, oh, we have to concentrate more on trying to save this species if there's trouble. But basically now it's no longer a common widely distributed species, right? So that's happening time and again with many groups. Now regarding what is the widely accepted species definition now, Kevin DeCouras proposed what he calls the general concept of species, where he has taken the various species concepts and put it under one umbrella. There are about 21, 22 different species concepts, morphological species concept, ecological species concept, phylogenetic species concept. I can go on, it gets very confusing. DeCouras said they are all talking about the same thing. They're all talking about, independently evolving metapopulation lineages. So you can use any of those species definitions to come up with your species. It doesn't have to be only morphology or only behavior or only genetics. Use multiple lines of evidence, and then you have greater confidence that you're actually dealing with multiple species. So that's where we are at right now. Yeah.
I just have one follow-up question, which is, according to this species, can different species mate with each other, successfully mate with each other?
Yes, according to the it used to be called lineage species concept. Now it's called the general concept of species. Interbreeding can occur. I mean, of course, if there's extensive interbreeding, then the two species will merge and you'll have only one species. But you can have hybrid zones. That's okay, right? But these two entities that you're calling different species exhibit divergence along other axes.
Right.
Right? They are actually found in ecologically distinct areas. Morphologically, maybe they are quite similar, but most other, and if you look at the genetics of these species, they are in fact different at most loci. And there's very little gene flow beyond the hybrid zone. Well, call it two different species. I mean, it's absolutely fine, you know. And often hybridization, what's happening is when we humans modify the environment, we bring in you know, sister species together, and because the environment is also very modified, they hybridize. So it might be an outcome of our, you know, messing around with nature. You know, often that's the case. There are natural hybrids also, though. Yeah.
Thank you so much. That was so interesting. So my next question is for Patricia.
Yeah.
So you talked about inbreeding, which is when populations are small and they end up mating with relatives, and it reduces the genetic diversity, right?
That's
so
right.
What kind of situations can lead to inbreeding? And, you know, is it happening more often nowadays? And also, what consequences does this have for wildlife populations?
So usually inbreeding occurs more regularly in small populations, just because there's not many mates to choose from. So the less individuals you have to choose from, the more likely you are to mate with your relatives. So this is why inbreeding tends to happen more often in smaller populations. In some species, inbreeding is actually a natural part of their evolutionary system or the mating system as well. But generally, inbreeding, when it leads to inbreeding depression, which is a reduction in fitness, that can affect many aspects of a species' life history, from its behavior, for example, in the black robin, which went down to one breeding female and obviously has very high levels of inbreeding. Females then started laying eggs on the rim of nests rather than within the nest itself. So there's behavioral aberrations that occur with inbreeding, for example. There's also physiological effects, so physical deformities that occur, for example, in the North American pumas. They have tail kinks and they have smaller testes and lots of physical abnormalities that occur with inbreeding. Also, there are reproductive issues and survival issues. So, for example, in the species that I work with, the hihi, they have hatching failure rates of around 30%. So 30% of eggs don't hatch. So they have high levels of early onset of mortality. So there's high levels of embryo mortality. That's quite common in inbred populations. So normally for birds, around 10% of eggs fail to hatch, but for threatened populations of birds, normally it varies between 20 and 80% of eggs fail to hatch. And that's mostly because of inbreeding depression, because a lot of the recessive deleterious alleles are expressed early in development. So all those alleles that are really important to the formation of that embryo are developed early on or are expressed early on, and hence the embryo fails to develop. So inbred populations tend to have higher mortality at early stages of development. So
in your work, you also mentioned a reduction of adaptive potential as a consequence of inbreeding or as a consequence of low genetic diversity. Can you explain what that means?
Sure. So measuring adaptive potential in wild populations is hard because basically, as I said before, you need really long-term data sets. You need to have pedigrees, you know, so you need to know the genealogy of that population, of if you want to call it that. That's what a pedigree is. So we know the parents and the grandparents and the great-grandparents of that individual. And the way that we use these pedigrees is we use molecular data to confirm them. So we know who the mother and father are. That's one way of using the molecular data. Or we can now use genome-wide molecular data to assess the levels of genetic diversity in the population. So in the hihi, we use the pedigrees to look at the heritability of traits, and that tells us whether that trait gives us an indication, it's a proxy for adaptive potential. So the heritability of traits are important for fitness, like reproduction and survival. So if those traits are highly heritable, then the population is more likely to have to adapt and survive. And also if it has more genetic variation, if there's more additive genetic variance in those traits, then they're more likely to adapt to changing environments. So we need to have high levels of heritability, high levels of additive variance, and also high levels of genetic variation in the population for that population to adapt to new conditions. And so we've measured all of these traits, all of these molecular markers in the hihi, and we found that unfortunately we have very low levels of genetic variation. So compared to other species where there's been genomic information is available, or the wild populations where genomic information is available, in comparison to those, hihi has very low levels of nuclear diversity and genetic diversity in general. But also we have very low heritability of those traits that I said were important for adaptation, like reproductive success measures, and also there's very little additive genetic variance in those traits in morphology or in reproduction. So unfortunately, from a genetic perspective, hihi has very low adaptive potential. But despite having very low levels of adaptive potential, hihi populations are still growing, and we're still able to establish them successfully in different parts of New Zealand. So there are other aspects of the species life history that allow it to thrive, for example, its plasticity or demographic changes that allow it to still thrive despite its lack of adaptive potential. But it means that there's a genetic risk that needs to be considered when managing the populations.
So why do you think it has low adaptive potential?
Again, because of, well, initially because when Europeans came to New Zealand, they changed the habitat, they brought invasive species, and so a lot of the hihi from mainland New Zealand disappeared, or they completely disappeared from the mainland, and they were left in one small population on an island called Hauturu. And so this was the only population of hihi left in the world, which was relatively small, around 2,000 individuals. And then in the 1980s, the reintroduction program started, which took individuals from this population and started new populations. And this created a founding bottleneck event, which means because you can only take a few individuals, they're not representative, they don't represent the genetic diversity of the original population. So they go through a genetic bottleneck. And this accelerates this issue of reduction in genetic diversity and then enhances inbreeding and inbreeding depression. So hihi has gone through multiple founding bottleneck events and hence has lost genetic diversity, and inbreeding has increased across all of that establishment history. And so the management interventions that we do have genetic repercussions as well that we need to take into account when we do them.
So this is obviously a very kind of special case because it was a reintroduced bird. Mm-hmm. But let's say you have any species which is threatened because of habitat loss and its range is declining, or if, you know, for example, the tiger populations in India are very isolated and they're very small. So do you think that they also would have like less adaptive potential?
It depends on the species. Like measuring adaptive potential is very, very species specific. And there's many aspects of that species life history that determine whether they have adaptive potential, they have genetic diversity or not. For example, if populations grow relatively quickly, they manage to maintain a lot of genetic diversity, while if they grow slowly, they tend to not. It depends on the mating system as well. So if you reproduce relatively frequently and you have large numbers of progeny, then maintaining genetic diversity is easier than if you're a slow reproducing individual or population. And so those are the variables that you have to think about when you're looking at these kinds of fragmented populations. But even in these cases where you have small or declining populations that are fragmented, I think initiating processes where you can increase gene flow between populations is really important. So creating habitat corridors or artificial immigration and immigration that really helps maintain genetic diversity. But obviously we need to understand these populations before we start implementing those management strategies, because there's other considerations other than genetics before you implement them. For example, disease control and all sorts of other things.
Thanks. So my next question is for Praveen. So your lab, you study, you use genetics to study the evolutionary history of many species groups, and you also study biogeography, right? Like from where a certain group has originated. So what, in your work, how do you think this is relevant for wildlife management?
Oh, okay. See, the problem when it comes to assessing diversity, we use like the more sort of traditional sort of view, just count up the number of species, right? One area has more number of species, so it has higher diversity, but we don't look at how those species are related to each other, right? So the next level of sort of characterizing diversity is looking at phylogenetic diversity, right? So you can have two patches having the same number of species, but one patch has species that are more distantly related to each other than in the other patch. So, the patch with the more distantly related species has higher diversity. To me, the third level of characterizing diversity is to look at biogeography. Where did these species come from, right? So if you take the history of India's biota, as I said, India was part of Gondwana, it drifted apart and then merged with Asia. So India does have some Gondwanan elements, though not vertebrates. Fishes, we don't know what's the story. But I'm sure that there are many invertebrates that have Gondwanan origin. So that's one component of India's biota. And then once India merges with Asia, you have elements coming in from Southeast Asia, from the Palearctic region, from Africa. So you have all these species with very diverse evolutionary history, right? Now, somehow we should try to incorporate the evolutionary histories of all these species when we are characterizing diversity, right? So I think that should be the next step, because if you have a patch which has species from diverse has species with diverse origins, geographic origins, to me that is a more diverse sort of patch than a patch where you have species only from, say, one biogeographic realm, right? So to me, this is informing us about the diversity from a completely different standpoint. So we have to move away from just looking at species richness to move and get on to the higher levels of diversity. So I think that's where I'm coming from. In fact, in Western Ghats, you have all these isolated forest patches that consist of these elements from Gondwana that we don't find anywhere else, and these lineages share their past with species in South America and Africa and Seychelles way back in time, right? The Nasica batrachus, the purple frog, is a classic example. It's that lineage has been around on India landmass since Jurassic, when dinosaurs were roaming about. Most other elements came in more recently, right? So I think this is something unique about some of these elements, which we'll understand only when we do biogeography, Yeah.
So this leads up to my next question, which is, you're saying that it's important to preserve diversity from many different aspects. You should also look at biogeography and try to preserve that biogeographic history.
Yes.
So is there any advantage to preserving these different kinds of diversity over just preserving number of species? Or even like if you have one species of bird and there's many individuals of that one species, why is it better to have multiple species over having one species? Is there any benefit for in general? Patricia, do you want to go first?
Well, a more biodiverse ecosystem is a more resilient ecosystem. Each particular species is a piece in the puzzle. It works within the ecosystem, and if the more pieces of that puzzle you remove, the less resilient it becomes and more unstable it becomes. And so you need diverse ecosystems. You need diversity in number of species and types of species in order for all the different ecosystem functions to happen and for it to be a healthy environment. So that's why you need diversity in general for resilience.
Do you have anything to add, Praveen?
Yeah, I completely agree with Patricia on this. But taking it along the same lines, ecosystem function for we humans, ecosystem services, right? These forests and these grasslands provide us with whatever clean air, clean water, pollination, what have you. So an ecosystem that where the diversity has been maintained will provide us much better ecosystem services, will provide us whatever optimal ecosystem service that ecosystem can provide. Now, if you reduce the diversity in that ecosystem, automatically the services it provides will also go down. Right? It will also make it less resilient, as Patricia rightly pointed out. Let me just give you a very simple example here. And this is, I don't know if this is published. I hope this is published. So I was talking to someone who works in the Western Ghats, and he's been looking at, you're talking about single species versus multiple species. This sort of ties in there. He was looking at streams that flow through, forested areas. If you compare streams that flow out of, hilly regions which has the native forest untouched versus hilly regions where forest has been cut down and then you have monoculture of whatever has been used for afforestation, mostly exotic species. Now, if you compare the streams from these two patches, turns out the streams that emanate from forests that are native forests remain. They are not ephemeral. They remain. They keep flowing throughout the year. I'm forgetting the word here. Anyway, so whereas the
Perennial.
stream, huh?
Perennial streams.
Perennial streams. Thank you. Yes. Whereas in the other patch, the streams go dry in the summer, right? So water is so important. The classic example of ecosystem service, which is heavily influenced by the number of species. Right, and we can keep talking about so many such examples, and people are beginning to realize that, and so yeah, that's my short answer.
Yeah.
So I'll ask you guys one last question, and then we'll move on to audience questions. So my final question for both of you is, how can we, inbreeding is a huge problem, and the fact that we're losing biodiversity and small populations, we're losing genetic diversity. So how can these things be mitigated? Like from your work, from all of your experience, do you have any kind of practical considerations that can be applied to wildlife management? Patricia, do you want to go first?
Yep. Well, obviously, the first things to consider are the main drivers of extinction. So habitat loss, invasive species, all of the anthropogenic threats that species currently face. Those are the first things we need to tackle before we tackle anything else. And so if you have larger, more natural habitats, less fragmented, less polluted, that obviously helps reduce the risk of extinction in most cases. And then if you do then have small populations despite all these things, and you need to manage that, then you can mitigate by using genetic management. So as I said before, starting thinking about genetic starting gene flow between the populations, creating habitat corridors, creating reserves, population reserves, for example, for the Tasmanian devil, which I was talking about earlier. They have a kind of a safety net population on the mainland in Australia that doesn't have cancer. And so maintaining those populations healthy and diverse is important if the other populations in Tasmania go extinct, so that they can then have a source of individuals to reintroduce. So those kind of literal management applications can help once the species is already in a pretty dire level. But before we get to that point, maintaining, healthy habitats is the key, really.
And from your work with reintroducing species, are there any like best practices for that?
The best management guidelines are provided by the IUCN for reintroductions, and anybody that wants to do reintroductions should definitely read those and be familiar with those. But one thing I've learned from the work that we do is we need community engagement. We need to have everyone on board. There's lots of different stakeholders in reintroduction, and they all have to participate and be happy for reintroductions to take place for them to be successful. And there's many examples where that hasn't happened and reintroductions haven't been successful because of it. So we need to have people on board for these things to work.
Thank you. Praveen, do you have anything to add?
No, not really, because she's pretty much covered everything, and that's not really my area of expertise either. But yeah, I completely agree with a lot of the solutions that she put forward. With this corridors to connect isolated populations, I think a lot is now finally going on in India. So hopefully it will help.
Yeah, I think there's many countries that are now starting to understand the importance of this. In New Zealand as well, they're trying to remove predators, mammalian predators, rats and stoats from the mainland of New Zealand as well, to try and recreate the habitats that they lost. So yeah, there's a lot of work going on to try and do that.
And for that, you have to understand kind of the past better.
Absolutely, yes. You have to understand the historical context of each of the species and the community of species in order to be able to introduce the right measures to save the species.
So one question that I got from the audience, which is where do you draw the line at describing different species that have very similar characteristics? And they've given the example of warblers in the northeast of India because they're so similar to each other. So are they really genetically diverse enough to be individually classified?
I guess I'll take that question.
Yes, I wouldn't know.
Yeah, so that's a classic case of cryptic species, right? And I'm sure they have very different calls. And they probably forage in different parts of the canopy. So temporally and spatially, they sort of are separated. So they're not really competing with each other, perhaps. So one has to really look into it more closely. Just morphology as a single axis to describe species, we need to move on, right? So if you think that they might be two different species, then look at these other axes, ecological axes, behavioral axes, genetics, use multiple lines of evidence. Yeah, so that's the short answer to that.
Another question is, considering that genetic diversity is so closely linked to biogeographic history, is there any benefit in studying species beyond current political boundaries? And is it logistically feasible to do that?
I mean, species don't have political boundaries. So that's probably just, species that don't occur necessarily across where we've delineated political boundaries, so that it doesn't really apply. So you just have to study species wherever they are. And hopefully politics won't interfere in that too much. But
Yeah, political boundaries are man-made completely.
I completely agree with Patricia on that. You just have to find ways to get around issues of political boundaries, collaborations with other researchers, and it's very long and tedious, but there is no way out because otherwise your work is incomplete.
Yes, absolutely. Yeah, dialogue, open lines of communication. And one of the important things that we do as scientists is communicate to the public and to create these areas where we can have open dialogue so that we can protect species better by creating these areas where we can all talk to each other and come up with solutions that benefit everyone.
So following up on that, someone is asking, is it challenging to communicate the specifics of genetics or such topics to policymakers and to government officials and to the general public? And how can this information be more accessible and less jargony?
Yeah, I find it hard. Yeah, definitely it is hard. But I think that's one of the things we have to work on as scientists, is trying to communicate more effectively to every kind of audience, to the general public and to policymakers, because otherwise our work just stays in the scientific literature and is never applied. So communicating to the public, communicating to policymakers, communicating to community groups, to indigenous groups, to anyone who is a stakeholder in species recovery or in threatened species in general, is important to bring them on board and be able to explain the work that you do clearly and concisely and in a way that's informative for everyone. But it's not easy to achieve. That's definitely true in my case.
So another question that's come up, which I think, Praveen, if you can answer, would be great. Do you have any views on the proposed cheetah reintroduction program in India?
I don't know why they want to do that. and that too, they're going to get African cheetah, right? So I frankly don't see any point in doing this. Why not just instead use that money to, reintroduce the Asiatic lion in other parts of the country? I think cheetah was already on the way out, and I don't know if now in such a fragment, there are hardly any grasslands also left, right? So I don't know why they are doing that. And CCMB at some point in time wanted to clone the cheetah, and I don't know what. It's such a waste of money. That's my personal opinion.
I mean, I don't know the reintroduction program that's attempting this at all, so I can't. I'm not well versed in it enough to comment, but it does seem that there's many other species and projects that are likely to benefit from this kind of investment.
Since you work on reintroduction, how can we decide when money should be invested into reintroduction and when it shouldn't? For example, with the it was a very planned and it coordinated reintroduction program. So when that decision was made, how can you make a decision like that?
The reintroduction program for was funded by the government, so it's centrally decided, and for each species has its own recovery group, which is made out of different stakeholders. So there's the government, there's the local community groups, because in New Zealand as well, we have iwis, which are the local Maori tribes, and they have a very strong say because are the cultural heritage of Maori. And so they have to be involved in all decisions relating to the reintroduction management of the species. And obviously, all the different sites that are involved also have their own representatives, and they have scientists like me and John Ewan and Anna Sancha and many others that support them in the species biology and genetics and other aspects of reintroduction. So it's managed from lots of different angles, and it requires a lot of coordination across different stakeholders, and people have different aims, and so we need to kind of try and coordinate all of those things. In terms of investment, prioritization is a really difficult one. It depends very much on local values and what people want and what people are willing to accept. You know, there's many different aspects to it. For example, in the UK, reintroduction has been quite topical because, for example, reintroducing beavers has been quite difficult. Reintroducing, they've talked about reintroducing lynx or reintroducing the white-tailed eagle. That's been quite controversial because a lot of people are scared of reintroducing these animals. So you have to have people on board before you go forward with these reintroductions, before you invest a lot of money, and also the vulnerability of these particular individuals. Animals they're really important individuals, so you shouldn't be embarking on a reintroduction without being very careful first that you have all your things aligned. I think.
Thank you. Yeah, that makes complete sense. So on one question, which I think is maybe a little bit inspired by Jurassic Park, is should extinct species be revived? Like species that have gone completely extinct, can they be revived and should they be revived to restore ecosystems?
I don't know if extinct species can be revived. If even if we have the complete genomes of, so people are able to now get complete genomes of organisms from subfossils also. So not subfossils maybe, but from animals that have gone extinct long time ago, and the bodies preserved in permafrost or whatever, they're able to get complete genomes. But the sequence, right? But I don't know if we have the technology to now take that genome and somehow make a living being out of it. I hope not. I mean, that seems like a very, very dangerous prospect. Extinct species, if the species has gone extinct long time ago, right, 10,000 years ago, it makes no sense to have it back. If it is something that has gone extinct very recently and played a very important role in it was an apex species in the ecosystem, and then maybe there is some value to trying to bring it back. But it's again like reintroduction, right? And as Patricia pointed out, there are many issues. And do we have the habitat for that? What is the local perception? Is it worth investing all that money? So the most important point is, what role does that species play in the ecosystem? And by bringing it back, will we be able to revive what was there earlier? If we can't, I see absolutely no reason to do it.
Yeah.
Yeah.
That makes sense. So just one final question to wrap up this webinar, which was really interesting. So what future directions of research do you hope to see in your respective areas of work? What research do you think we need, and that's missing?
So from my perspective, I think one area of research that we need a lot of work in at the moment is female reproduction, actually. So we have a lot of understanding of male reproductive failure, and because across the last few centuries, men have dominated science, there's a lot of work on male reproduction, but actually there's not enough work on female reproduction. And so for most taxa, we understand very little about why female reproduction fails. And when it comes to helping threatened species of animals, we need to understand female reproduction in order to enable us to reproduce them successfully. So I would make a call of arms to start working on female reproduction and female reproductive failure.
I'm surprised that that's the case even in wildlife, that male reproduction is studied more.
Yeah.
I thought it was just humans.
Yeah, hugely biased. Yeah.
What about you, Praveen?
So, of course, I use genetic tools to understand diversity. So, I would say, yes, it is, we should be doing more of it. There's so many groups to that needs exploration. So definitely this is some area where we are slowly building up, but not enough, not fast enough. But I would quickly like to highlight the problem of how taxonomy as a field is dying, right? And that is a tragedy. I think we need to do something to revive taxonomy and make it cool and sexy and fashionable, because that is the bedrock of biology. Every field in biology, ultimately, you're working on something and you need to identify it. But in colleges across the country, there are hardly any taxonomists left. How are we going to characterize all this biodiversity? We can just sequence stuff and not be able to identify what it is, right? So I think we should definitely go the genetic way that is needed, but along with that, go back to the basics, encourage taxonomists, make sure they have opportunities.
Yeah,
Wow.
I agree.
Thank you so much, both of you. It was such an interesting webinar, and I feel like I have a lot more questions that I would ask you if I had the time. But since we've run out of time now, I would just like to, thank you very much for participating, and thank you to our audience for attending the webinar. And yeah, I hope to see everyone in the next edition of the CWS Wildlife Chronicles. So thank you. And
thank you for inviting us.
Yeah. Thank you.
And yeah, thank you. You can obviously look up these two speakers if you want to read more about their work. And yeah, thank you so much.
No problem.
Nice meeting
Bye.
you.
Yeah, lovely meeting you. Take care.
Bye.
Bye.